Good morning, everyone. Thank you, Alain, for the invitation. It's a real pleasure. So I'd like to continue on the theme that Maria introduced, that of morphogenesis. And the title of my talk addresses what I think is an interesting and, in a way, to some extent, surprising new way of thinking about tissue morphogenesis, which stems from unpublished work, and I will just present some aspects of it today for your thinking. And that is indeed the control aspects and the self-organizing properties of tissues. So I'd like to start with a simple image which captures, I think, the fascinating, important theme that my lab and many other labs are working on, which is trying to integrate the information aspects of morphogenesis, which addresses where, when, and along which direction cellular events occur, as well as the mechanical aspects, which really addresses the forces that are at play at different scales, and what are the material response properties of cells that will dictate, indeed, collectively, the length scales and the time scales of processes. So biochemical information, the morphogens that several of you have talked about, and mechanics share the property of defining length scales and time scales. And I think that maybe to enrich the theme of morphogenesis and morphogens, it may be important to consider as well the way that mechanics actually dictates deformations, and in particular, as well as the morphogens can diffuse through a tissue and define different domains. Likewise, deformations can diffuse through a tissue depending on the mechanical properties. So in a way, I think that the view that biochemistry and mechanics will integrate together to really dictate cellular response and tissue response is what I think we should be thinking about more, and it is definitely a direction that my language is pushing forward. So the important distinction that I would like to present today is that of orchestrated dynamics and self-controlled dynamics. And to illustrate that very simply, I have on the left a picture of a construction from Gaudi in Barcelona, where you could recognize the ideas that there's hierarchical control over the information that deploys at different scales to dictate cellular identity. But there's a different way of actually organizing very complex dynamics, which is self-organization, whereby I mean that simple local interactions are sufficient to produce very complex structures. And I think the very important distinction between orchestration on the left and self-organization on the right is that the amount of information that is contained to characterize this system is very different. On the left, you probably need many genes, many instructions, a whole series of interactions to orchestrate the way it is, maybe like an early fly embryo. I mean the patterning information in the early fly embryo, while self-organization, very local information, local interactions can be enough to produce very complex structures. So this is an interesting aspect. So while I will refer to self-organization in my talk, I will mean, well, there are local interactions which could be biochemical or mechanical, which collectively could produce interesting dynamics. So to extend the introduction on this orchestration, what I want to say and give examples is indeed there are many different ways there are a number of terms in developmental biology that are characteristics of this vision of development where things are orchestrated through genetic means. And I mean by that selector genes, for instance, master regulators, and even morphogens to some extent. They do contain the idea that a substance would have long-range effect and will induce a number of different properties by sort of the hierarchical, they will sit sort of on the top of the hierarchy of interactions. And the way that we understand morphogens and the regulatory networks that James probably talked about yesterday indeed convey this idea of a very controlled and top-down aspect of morphogenesis. And this provides me a means to further document the aspects of information in the early fly embryo, the anterior posterior information that is really important to convey cell dynamics along the AP axis, as well as the dorsal ventral polarity that Maria introduced to you. So now you will be very familiar with the fact that in the early fly embryo, there are two kinds of morphogenetic processes. The one that Maria talked about, invagination, taking place in the ventral half of the embryo, as well as endoderm invagination, which I will talk to you about in the posterior. And this is driven by apical constriction, and it's a fairly rapid process. I mean, the apical constriction on a few minutes time scales. However, if you look on the lateral flank of the embryo, there the tissue flows on a longer time scales, typically a few tens of minutes, and their cells undergo fluid flow, and you can see the cells actually spreading as if along the anterior posterior axis. They do not divide. They simply change their neighbor position. They intercalate. And these two processes are driven by active contractile processes induced by myosin II activation. So this is a slide that introduces the invagination that Maria talked to you about in apical constriction. I'd like to focus on another process, which is tissue extension, which is driven by the orderly remodeling of cell junctions, and in particular, a subset of junctions along anterior posterior neighbors shrink, produce then new contacts with the establishment of contacts between these two cells along the dorsal ventral axis. And the reversible deformation of cell junctions gives rise to a local extension which collectively produces the whole tissue extension. And this is driven by local activation of myosin II, which is not in the apical surface, just like Maria showed you, but rather preferentially along the edges of the junctions shown here in green. So here you have yet again summarized the idea that tissue geometric deformation, tissue extension, is driven by cell intercalation, and this in particular is characterized by topological change in the junction configurations, and this is driven by anisotropic stresses coming from planar polarized activation of myosin II. So indeed, spatial control over myosin II activation determines very different cellular deformations and tissue deformations. And if myosin II is activated apically, then isotropic tensions give rise to geometric transformation at the cellular level and topological changes at the tissue level. You produce an invagination. But at the, if myosin II activation is anisotropic, produces anisotropic stresses, then you induce topological transformations, it is cell intercalation, and give rise to geometric deformations at the tissue level. I mean by that tissue extension. So the cells have a means to control myosin II activation pattern, the polarity, and thereby the polarization of subcellular contractile stresses in the cellular level, and these have deterministic tissue outcomes. Now, this provides a fairly static description of contractile stresses at the cellular and tissue level, but there's a further features of contractile dynamics which Maria introduced to you, which is pulsatility, which is really important. In fact, both in the mesoderm, as shown when Martin was working with House, and in the ectoderm cells, the apical surface of these cells is contracting in a periodic fashion on an order of 100 seconds. And this is seen here on this movie. You see myosin II minifilaments that actually aggregate. And then after they aggregate, the whole structure collapses. And this is against a mesh of actin filaments, which you can see here down there. Now, the little network that activates myosin II, which is composed of the GTPase RO1, the kinase ROCK, and activates myosin II through this phosphorylation, the whole network is pulsatile. And interestingly, this pulsatility requires not only all the components, but also feedback from myosin II onto RO1 GTP, which is the activator of this whole pathway. So we have an example of a local circuit whose pulsatility is untrained by mechanics through the contraction of myosin II. So this provides sort of a landscape in which we can think now about how embryos control, how the information of embryo provides a means to control cellular mechanics. So cellular mechanics, what I mean today, is myosin II activation here. And what we need to understand is how is it that cells that are all born alike with respect to the composition, at least the number of cadherin molecules and myosin II, early on at the onset of gastrulation, within a few minutes, they undergo different pathways. In the mesoderm, the cells will activate myosin II at higher level, giving rise to sort of these, I draw it as a blue star in the central of the cell, and these cells would undergo apical constriction. While if the cells are in the lateral flank of the embryo, they do have a little bit of apical activation, that is pulsatile, but they chiefly have a lot of myosin II at the junction shown here in red. So two different polarity of myosin II activation emerge, and these are driven by a single molecule, which is Rho-GTP. Rho1 GTP activates ROCK and myosin II in a domain, in a pattern that is very similar to myosin II. And so what you can now see as a color-coded pathway is actually two branches of a pathway downstreams of G protein-coupled receptors, GPCRs, which actually activates distantly rho GTP apically and rho GTP at junctions. And I mean by that G alpha, one of the subunits of the heterotrimeric G complex downstreams of GPCRs, activates selectively RhoGEF2, which is an apical GEF required for rho1 GTP apically, while at junctions, it's G beta gamma that are required for this activation through a different GEF, which was recently discovered, P114 RhoGEF in mammals, which we call wireless for reasons which will be evidenced in the next slide, and that activates rho1 GTP at junctions selectively. So these two pathways provide a means to selectively control rho1 GTP in different cellular compartments. And what we do not understand is how is it that in the mesoderm and the ectoderm, the activation of the blue or red pathways are quantitatively different. So now I'm going to show you a piece of data where we just activate G alpha GTP or G beta gamma, and you will see the dramatic consequence in terms of myosin 2 activation. You have on the left now a still image of an embryo in the ectoderm. You recognize the planopolarized cables of myosin 2, which I described to you before, and you see sort of peckles here in the apical surface. These are just still images through pulsatile myosin 2 network. This is not background. If you just overexpress G beta and G gamma, what you see is dramatic hyperpolarization of the cells, where there's more myosin 2 junctions and there's very long cables spanning all the embryo, while you didn't change the concentration of myosin 2 in the apical surface. Conversely, if you just activate G alpha in the GTP lock mutant QL, you now increase selectively the apical activation of myosin 2, and the cells are confused as if they were undergoing apical constrictions. They actually don't, because it's a whole tissue that actually activates apical myosin 2. But clearly now we have put our hands on the two knobs that control junctional versus apical myosin 2, and these lie downstreams of GPCRs. So putting it back now in this context, the challenge is to now understand how is it that this single pathway that is very general could actually work, use developmental inputs, shown here in a color-coded fashion, to activate in the mesoderm more myosin 2 apically than in the ectoderm, and conversely in the ectoderm more myosin 2 junctions. And so here's the number of genes discovered by essentially Maria and Eric's lab that shows sort of the developmental inputs that provides a variation on the theme of GPCR control of rho activity. One is the ligand FOG. FOG is actually ubiquitously expressed. It is at low level in the ectoderm and very high level in the mesoderm and in the posterior endoderm. If you remove FOG, in every single cell in the embryo, including the ectoderm, blue activation of myosin 2 is gone. This has a very dramatic effect in the mesoderm. There's no tissue invagination. And in the ectoderm, you have no pulsatile myosin 2, and cell intercalation is also affected. There's another GPCR that potentiates GPCR signaling, and there is this very important molecule, ST48. It's a fly-specific molecule, as we know. It's just required to localize RhoGEF2 and increase its apical activation. So this is a good example of a developmental input, which you could think from a evolutionary perspective of how a single gene could just play on this pathway by just increasing RhoGEF apically, RhoGEF activation. And in fact, if you express T48 in the ectoderm, you do exactly as if you activate G-alpha QL or RhoGEF2. You selectively increase apical myosin 2 activation. So these are a number of developmental inputs that actually provide a means to quantitatively activate the blue RhoGEF2 pathway. Now, what about activation of myosin 2 junctions? Well, the very important family of genes that have been characterized by James Allen's lab recently, downstream of the parallel genes Eve, actually are essential for that, although we don't know the mechanism. These are expressed along stripes, and it's been proposed that the combinatorial expression of the surface molecules provides a means to break the symmetry of these cells. Our evidence documents that actually it's not so much the combinatorial activity, that actually single stripes of a single toll does break the symmetry, and they do so by activation of a GPCR, which activates G proteins. So there's a lot of things to do because there are many questions marks we don't understand, but I wanted to present to you sort of a working model of how we see a conserved pathway being activated quantitatively differently in different parts of the embryo to activate a contractility in different cellular compartments, namely the apical surface or at junctions. And I've shown you how differential activation of myosin 2 in these different subcellular compartments does have different cellular outcomes and tissue outcomes. So this closes a presentation of mechanics, which I think is very well illustrating what I meant by orchestrating mechanics through positional information in the embryo and playing on subcellular activation of a Rho pathway which controls cell contractility. Now, I would like now to switch to a system which seems to work partly on the same principles, but now also deviates quite significantly. And this will be the part devoted to self-organization. So this is a part of the embryo which has been less studied because it's present in the posterior, it's kind of roundish, and it moves pretty fast. But Claudio, a biologist in my lab, and a physicist, students, Anaïs, worked together. They wanted to collaborate from day one, and have done a great job of characterizing what is going on in the posterior half of the embryo. This is now a 2-photon slide through the embryo. We're not looking at it from the surface, because you can see a number of remarkable processes going on. First, a rotation with a given polarity that is always migrates toward the posterior, towards the dorsal part of the embryo. Then you can see an invagination, very similar to the mesoderm. And then you see something which will be the major focus of my talk, which is a progression of something going on here. So let's look at these cells now from the surface. Now you're looking at the dorsal part of the embryo near the posterior, and what you will see in green is activation of myosin II, and the cells are marked in purple with cadherin marked in purple. And what you will recognize to begin with is the activation of myosin II in a way very similar to what Maria characterized in the mesoderm. Namely, you have activation of myosin II in the apical surface, and this is, as we'll see in the next slides, driven by positional information from the posterior endoderm. But now you will see a number of cells in this region which do not have myosin II activation, but they will actually sequentially activate myosin II, as it is actually activated through the tissue in a wave. And you can see this cell activates myosin II, dives into the furrow, a new cell is activated later on. It goes very fast. There's a new cell that activates myosin II every 2.5 minutes. You have here the timescales on the top right here. So we define a primordium region where myosin II is initiated to begin with in a very specific pattern, and then a domain of propagation where the progression is linear. So here's a way of representing that. You have the primordium, here a Rho cell we mark, which does not have myosin II, but about 10 minutes later, it activates myosin II, then is incorporated into the furrow and disappears. But then the next row of cells, further away, another 10 minutes later, will activate myosin II and will be inside. So effectively, there's a very significant part of the dorsal part of the embryo that is eventually becoming activated, but instead of being activated all at once, as I've shown you in the mesoderm or in the ectoderm, it does it in a way of a traveling front that propagates through the tissue. And we'd like to understand how this is occurring. The progression is interestingly linear, as you can see here. This is here reporting the area of the cell, or here myosin II intensities in the tissue. You have here time, and here you have the anterior posterior axis. So indeed, here what we find is activation of myosin II in the primordium. Then this is a furrow, and here is myosin II activation linearly. This is not just occurring at the tissue level. If you look very carefully at any cell in this tissue, here's it blown up, you can see that myosin II is activated in a wave from the posterior of the cell and actually travels through the anterior edge of the cell. And here's this a chymograph. You have time and space. This is a single cell. This is the anterior boundary of the cell. The posterior boundary of the cell is actually moved to the left because the cell is stretched. And then as it constricts, myosin II is actually seen sort of activated sequentially towards the anterior. And this is a very unique dynamics that we actually do not find anywhere else in the embryo, where we have pulses and flows, but not this wave activation. This is not myosin II moving, it's new myosin II minifilaments that are activated in the cell by proxy. It's not just myosin II that is traveling through the cell, it's also its activator Rho-GTP. ROCK, the kinase, which I don't show, similarly propagates in a wave through the cell. So there's a whole network that I showed to you before, which actually is activated in the wave dynamics. So in the context of the embryo, there in the posterior, you have different maternal and zygotic inputs that control the very same pathway that I mentioned to you before, namely the ligand FOG, which activates the GPCR, the G proteins, myosin II activation, and apical constriction. The difference between the mesoderm and the ectoderm are just the transcription factors that actually activate the same molecule FOG. But we are in a very similar context for the initiation of the process. Indeed, so this is sort of a little diagram that shows you the activation pattern of the receptor tyrosine kinase torso, which induces transcription of heuchebine and TALES in these different domains, shown here in red and green. And if you just look at the torso, no maternal mutant, you can see that myosin 2 is not activated at all, and so the whole system is blocked. And similarly, if you have a strong knockdown of FOG with RNAi, we think there's a low maternal contribution of FOG, then you have a similar phenotype that you completely lose myosin 2 activation and have no progression. So clearly, the activation of the myosin 2 in the primordium is under dependence of the transcription of heuchebine and TALES plus FOG transcription, which activates the GPCR pathway. But that's so much for the induction. What about the propagation? And so a simple idea that we wanted to test is whether the production of FOG is diffusion from the source might actually serve as a morphogen, in the sense that the concentration would dictate the range of activation, and might it increase over time and diffuse and thereby produce sort of a wave of activation through the tissue. A simple prediction of this model, though, would be that the progression would be not linear, and in particular that the rate of activation of myosin 2 at the source or near the source and away from it would be different, and in particular that it would be lower as you move further and further away from the source. So we tested this idea by doing simple experiments, and the first one is just looking and comparing the dynamics of myosin 2 activation near the source in the primordium and away from it. And you can see here an example of a cell in the primordium, and here in the propagating domain. Best to see actually the dynamics of a single cell, where you can see pulses and this characteristic trace of myosin 2 in the primordium. In the propagating region, myosin 2 is a very low level and actually is induced very rapidly to this maximal level. Now, averaging many cells, this is the difference in the range, in the rate of activation. So clearly, if you're in the propagating region, the rate of activation of myosin 2 is greater than anywhere else in the embryo, whether in the mesoderm or in the endoderm primordium. There's something special going on in these cells here that allows them to activate myosin 2 very rapidly. So now we play with the dose of FOG in the source. There's a construct that Maria made, which is a heuchebine promoter expressing FOG. You express that on top of the endogenous, so you effectively double the amount of FOG expression. And clearly, you can see that comparing the two embryos that are timed together, FOG activation in the overexpressed FOG is occurring earlier. The domain of activation is slightly broader. Despite of that, the kinetics of propagation is completely unchanged. And I'm not going to show you, but we've manipulated the expression of FOG, even expressing it ubiquitously in the embryo. In fact, in this region, you still have a wave on a normal propagation kinetic. So the system seems to be, the propagation of myosin 2 is independent of the expression pattern or the dose of the ligand FOG. And here's some quantification, where actually you use, again, as a reference, a very simple diffusion model where if you overexpress the source, the ligand at the source, you expect to have an increased rate of activation with a decay over in time as a function of the distance from the source. And here, comparing the wild type in blue and the overexpressed FOG, you can see that if anything, the rate is actually lower, but it is not greater than in the wild type. And you see here that at time 0, there's indeed more FOG because there's more myosin 2 because the activation is occurring earlier, but the rate is not changed. And similarly, in the propagating region, the rates are not significantly different. So, in other words, FOG is important to time and to set the domain of activation in the primordium, but the kinetics of propagation is independent of it. So let's look now further and push this idea a bit further. FOG, you can detect it with an antibody. You can see it now first induced in the primordium, as you can see it here. And FOG subsequently in the furrow is well detected here, but actually not so much detected in the apical surface. There's a little bit of it we can sort of guess here, but it's chiefly present in the furrow and not so much in the apical surface. So we decided to look at FOG transcription dynamics directly. And the way to do it is using this MCP-MS2 system. This is a paper from Thomas Gregor's lab where they looked at Eve dynamics. You take MS2 stem loops, which we put in the 3' UTR of FOG. And if you express low expression of MCP, GFP, which recognize MS2 stem loops, then indeed it will mark very strongly the locus where the locus that actually is a concern, and in our case, that would be FOG. And if you have 2 copies, you will see 2 spots because it's deployed. So here I'm showing you still images of the embryo where we have here in the primordium the dots of FOG transcripts production, and you could see it on the side, and here actually using a marker of the nucleus with a histone RFP, you can see clearly the dot on the side and on the top in the primordium. Later on, in the propagating region, there's absolutely no FOG detected in this region. So not only don't we detect high levels of protein, but the transcripts is not made there. So FOG cannot be, the transcription of FOG cannot be the cause of the dynamics of activation of myosin 2. We decided to push this even further. Can we just block transcription altogether and see whether the dynamics of myosin 2 activation is still occurring or not? And so this is an experiment that involves the injection of the very potent RNA Pol2 inhibitor alpha-mannitine. If you inject alpha-mannitine in the middle of cellularization, well, you will block FOG transcription, and you would have no mesoderm and no posterior endoderm activation, and you will block everything. But now if you do it slightly later, at the end of cellularization, once we believe FOG production has occurred, then what happens? And so this is shown in the same movie, in the next movie. You see on top a control embryo, and at the bottom, an alpha-mannitine injected embryo. Oops, there's a movie, an important movie to look at. Well, you would see that the race is going on, and they arrive at the same, and they move at the same speed. Now, we know that these embryos do have a strong phenotype, which you expect from alpha-mannitine injected embryos. For one thing, there are these mitotic domains which depend on acute transcription of CDC25 string, which produce these mitotic domains here. They are gone in these embryos. So we know that string is not transcribed. We also know that TOLs and EVE, which are required for planar cell polarity, which give rise to these cables of myosin 2 here in the control, are not occurring in the alpha-mannitine injected embryo. So we're very confident that, yes, indeed, alpha-mannitine has a very strong effect. So we concluded from this experiment that clearly the dynamic, the progression of myosin through the dorsal epithelium is certainly induced by transcription in the primordium, but is completely independent of further transcription, and it has to be dependent upon another mechanism. How many more minutes do I have? 5 minutes? So, I mean, I like the idea of falsifying an idea, because we know it's not what we thought it was. Now, I would like to present to you the current status of our understanding of what we think it might be dependent on. So, the idea that came to mind was that there was some kind of mechanical feedback that would be acting from the primordium and being required for the propagation through the dorsal tissue. So, a first way to test this idea is to do some laser ablation experiments and ask whether the cells here are under tension. So we do a line cut, and we look at the relaxation kinetics, which is a good indicator of the local tension that is in the tissue before you cut it. So you could cut along this direction or along that direction. And you can do it near the primordium or away from it. If you do it away from it, cutting in both directions gives similar results. There's no polarity in the tension. However, if you're close to the primordium here, there's an increased tension along the AP axis, the anterior posterior axis, compared to the lateral axis, as you can see here. If you are in a torso mutant, where there's no activation of myosin to the primordium, then in this region, you have no difference in the tension in the cells. So these cells are undoubtedly under polarized stress coming from the primordium that is pulling on them. And in fact, you see they do have an elongated shape. Now, this next slide is a very important experiment, because we tested the idea of a mechanical feedback by doing the following. I'm not showing the slide, I'm explaining the experiments before. So we know that rho GTP, which we report with a biosensor, and myosin 2 propagate in a wave at the same dynamics through the tissue. And we're going to inject an inhibitor of myosin 2, a ROCK inhibitor, Of course, myosin 2 will disappear because it will no longer be activated. But we'll look at raw GTP biosensor and ask, is it continuing to propagate through the tissue, or does it stop? And you would see, so this is the experiment. The top you have a control, and you can see myosin 2 and the raw sensor that are activated and propagate through the tissue. And at the bottom, let me show it again, you have myosin 2, you inject, so it disappears immediately, and you would see that the raw sensor is also inhibited, and its characteristic high activation and propagation is gone. So let me show it again. They begin at the high level, similar to the wild type. You inject, you immediately see a dilution of the raw GTP sensor, and you no longer have its activation. All you have left is a very low presence, which is not noise. This is dependent, we think, on probably low fog expression or presence that actually poisons the system. But the transition to a high activation and its linear propagation is dependent upon ROC activation, myosin 2 activation by ROC. So there's a feedback of activation of raw GTP upon by myosin 2 from myosin 2 activation. So what is the nature of this feedback? Is it stress per se? Is it the strain that it induces? Or even can we consider the geometry, namely the curvature of the tissue, which could be consequence of the stress that emerges from myosin 2 activation here? So all of these could potentially play an important role in this feedback. And let me first describe an important geometry and changes in geometry at the whole tissue level. At the beginning, we were thinking in the 2 dimension, and actually my movies were essentially looking at the surface of the cells. But we think that the proper way to understand what is going on requires looking at the tissue in 3D. And so what I'm showing to you here is a 2-photon slide through the whole dorsal epithelium. And what you will see, so the movie is looping so I can comment on different features. These cells here are columnar. You will see gradually that this is, as this invaginates, the cells are pushing against this piece of the tissue. And indeed, the cells which initially were vertical in this image will bend because they are pushed by these cells here. So these invaginate, they push here, and you see the high curvature of the cells. Okay, so I wanted you to notice that the cells are being pushed by these cells here. And what I will document in the next few slides that as they are being pushed, they are being also lifted against the roof, so to say, which is the vitelline membrane that encapsulates the whole embryo. So I'm going to describe what is essentially proposed to be a mechanical cycle whereby cells are being pushed by the invagination behind them. As they are pushed anteriorly, they're also lifted toward the apical against the vitelline membrane. I'll show you in the next few slides. And so let's look now at what happens here in the next few slides. Now, to better document the bending of the cells, here you view from the top at 3 different times a cell shown here on the side. You can see here it's vertical, it's not bent at all. And a few minutes later, you see that it's 2 apical junctions gathering. One is already moved apically, the other one not yet. And the cell is already bent a bit, and later on it's bent even more, completely curved. So we consider the possibility that the cells could be actually moved against the vitelline membrane as a consequence of this pushing force. If you have a tissue and you produce an invagination, you expect and see in computational models that there are actually lobes around it, which is a consequence of pushing it. It would produce some kind of extrusion outside. So this would indeed push the cells against the vitelline membrane. So what NIS did is he injected fluorescent dextran in the vitelline space. That labels this whole space here, and you can see the outline of cells, and there's a strong signal. Here you see the strong signal in the invaginated area, but what you can notice is that the cells that are right ahead of the furrow, as myosin 2 is activated in green, are devoid of dextran. So the cells are being pushed against the vitelline membrane, and they indeed exclude completely the dextran. So that suggests to us that maybe there is actually friction going on with the vitelline membrane. So that is actually quite clearly seen in these cells, which are as if stuck and then rapidly relax. Here what we're looking at is myosin 2 as they contract. initially you see them forming this sort of boundary. One is sort of stuck behind and will rapidly detach. You can see it better also on the side. You will see here the cell is stuck and snaps back after it detaches. So clearly, what I want to say that the cells right ahead of the furrow, right in the region where myosin 2 is activated, are lifted against the vitelline membrane specifically, and are undergoing high frictional coupling with the vitelline membrane. We don't know at all the mechanism, whether it is just passive friction or actually specific adhesion. We actually can see that more clearly when we look at the meshwork of actin filaments in these cells. So here you have one cell, and what you have in yellow are references in the microscope or in the embryo. Consider them as static points, and the cells are going to move through the tissue because they are pushed anteriorly by the posterior endoderm. As they do so, they will spread, and if you look carefully at the actin speckles, you will see that they jiggle around, but at some point they will become immobilized in the referential of the embryo. So the cell is being moved towards the anterior. It spreads because of its being pushed and stuck to the apical surface, as if it's glued, and these speckles now are immobile. They jiggle around, but they don't drift toward the anterior. So you can see it in a chymograph much better. You have time here and space. This is one cell that we looked at here. The cell, everything is sort of drifting toward the anterior at the beginning, but at some point, these little speckles of actin meshwork jiggle around, but they no longer move together with the cell. This is a movement of the cell edge here, but the actin filaments are no longer moving. So that is actually indication that the apical actin meshwork is being frictionally coupled to the apical vitelline membrane. So think of the cell is being moved anteriorly, but because it is actually glued as if to the vitelline membrane, the actin meshwork is actually immobilized. So this is an important element in the mechanical cycle that I will present to finish now. So this is a first sort of a multicellular description of an important aspect of the mechanical cycle. The cells here are being pushed by the invagination, as we see. In doing so, they are lifted up against the vitelline membrane to which they are frictionally coupled. And the friction here is associated with a long spreading of the cells that are immobilized. And at some point, these cells detach, contract, and we think that the contraction of the whole cells here in green is part of the mechanism that produces the progression and the pushing force toward the anterior. So you don't need to invoke any specific molecule here beyond the fact that there is contraction that draws the cells inward, pushes the cells, lifts them against the vitelline membrane to which they are frictionally coupled. And something is going on here which is critical, which is this activation of myosin 2. So what I'm going to say now is present a working model of what we think is going on in this context. We've done many manipulations, which I'm happy to present later on, but I don't have the time in the context of this presentation today. So the working model we have is one in which you have sequential 3 steps in the activation of myosin 2. First, we begin with a FOG, GPCR-dependent, low activation of myosin 2, which is seen in the form of little speckles of minifilaments activation. The cells here, the apical cortex is activated. Now the cells is being pushed toward the anterior, and it is actually frictionally coupled to the vitelline membrane, as I showed you, and that gives rise to a spreading of the whole cortex. So next step, the junction will be both moved apically. But once the cortex is moved apically and the junctions are stuck there, contraction of this meshwork is going to be resisted by the frictional coupling to the vitelline membrane and the coupling to the junctions, and that gives rise to strain-dependent stabilization of myosin 2. It is very well known that motors, and myosin 2 in particular, is activated by strain in the meshwork that stabilizes myosin 2 motors by playing on the duty ratio of the motor. So from a low activation triggered by FOG, we have a strain-dependent stabilization whose critical element is actually the existence of frictional coupling to the vitelline membrane and to the adherence junctions. And indeed, if you, for instance, knock out alpha-ketinin, which is required for this coupling, then you no longer have this high activation and propagation through the cell. But this is just a second step. The third critical step is that further pushing the cell, you have a detachment of the cell from the posterior, which gives rise to a rapid de-adhesion and collapse of the actomyosin network and give rise to this maximal activation of myosin 2. So moving through the 3 steps require the pushing forces in the black arrow here. When we do the ROCK-inhibited experiment, we block this altogether. So the whole the compression that emerges from myosin 2 activation of the primordium is an essential element for the progression from low activation to strain-dependent stabilization and eventually to collapse. And the system, as it collapses, provides a recycling of the whole system because these new cells will participate in the furrow and will allow the compression to progress step by step. So I'm finished, and I just have these concluding slides where I just present to you a good example of where we think is a genetically programmed activation of cell mechanics that activates myosin 2 through the GP cells and Rho signaling in the primordium. Its subsequent dynamics and its linear progression is not even dependent on transcription, which is kind of odd in a fly embryo, sort of probably the paramount example of a system whose morphogenesis is highly controlled by genes controlled in specific domain that control mechanics, as I showed you, and has been talked about by many people, and is clearly very important. But there are some aspects of morphogenesis which do not respond to this kind of, follow this kind of scheme, and seem to depend on feedbacks that are mechanical in nature. And I finished thanking the people involved in this work. Anais and Claudio have done all the experiments, and Anais also done some modeling. Together with Ed Munro, our collaborator. I didn't talk at all about all the simulation that she's done, but the last model that I presented is one that is actually strongly supported by simulation with Madan. And I thank you very much and take any questions.